Sample 2, again, copy the question down in your worksheet. Sickle cell anemia is an autosomal recessive disorder that leads to sickle-shaped red blood cells that block blood flow and do not carry oxygen very well. What are the probable offspring from a cross between a carrier female and a carrier male? Again, pause the video and copy that question down. Continuing on now. Well, we know it's autosomal recessive because they told us right there. If they didn't say anything, we would know it's autosomal, and to figure out recessive or dominant, you would just keep reading. We know it's one gene because we're talking about sickle cell anemia and nothing else. So we know it has 2 alleles, monohybrid, and 4 boxes again. So what are the alleles? Well, sickle cell is recessive, so whatever I have to designate sickle cell to be on the lowercase letter, whichever letter I choose. Doesn't matter what letter I choose, just I have to put sickle cell on the lowercase letter. This time I used N. I don't know why, but just chose it. I guess N I meant to say normal, but healthy. And again, little n is sickle cell anemia because it's recessive. They told us the disease is recessive. Now, we go with the parents, and here they said that the parents are, the male is a carrier, and the female is also a carrier. So to be a carrier means that you don't have the sickness. You're not sick, but you're carrying it, so you're hiding it. And so the only way for that to be possible is to be heterozygous. So big N, little n, big N, little n. Again, I then find the sex cells. It's one gene because there's just the letter N, one letter. So I go ahead and segregate. And then they also said we have to go ahead and make the Punnett square, set it up. So I put the sperm there. I put the eggs down there. Then I fill out the offspring, again by fertilizing sperm and egg, down and across, down and across, down and across, down and across. And by looking at this, it looks like these have the dominant in front, therefore it's healthy. So 3 out of 4 are healthy, and 1 out of 4 has sickle cell anemia. Sample 3, write this one in your notes. Color blindness is a sex-linked disorder where one cannot distinguish certain colors from others. What is the probability that a colorblind female is born from a female carrier and a non-affected male? Again, we look this time they tell us right away it's sex-linked disorder. So it's sex-linked, and it's recessive because color blindness, or most sex-linked disorders, are recessive. It's one gene, 2 alleles, and monohybrid, 4 boxes because we're only talking about color blindness. Big C is healthy, little c is colorblind because little c is recessive. The colorblind is recessive. We figure out the parents, and here they said that the male is not affected, the female is a carrier. So for the guy to be not affected, well, we know we have to use X's now. It's not autosomal anymore. It's not where we just put letters. We have to put the X's or designate the sex chromosomes because it is sex-linked. We know guys are XY, and we only put stuff above the X because it's X-linked. And so we put big C because they said the guy is not affected, so he's healthy, so big CY. And the female, which is XX, she's a carrier, so that means she must have a big C and a little c. And then we go ahead and make the sex cells, and again, there's only one gene we're talking about. We're talking about color blindness, so we just separate. Separate. Then we figure out the Punnett square, set it up. Again, put the sperm there, put the eggs over here. And then we go ahead and fertilize to get the offspring. And again, that's how we do sex-linked traits. Notice it's different. We use X's and Y's or X's and X's with the superscript. That's for sex-linked traits. And then we could go ahead and look at this, and what we find are that 0% are colorblind. They want to know if there's a colorblind female. And I look at the females. Only look at the females here when they ask these types of questions. Or in this case, they're asking how many are colorblind females. So I look at the females. Both of these are healthy. Or this child is healthy, sorry. And this child is a carrier, but she's healthy, so they want to know how many girls are colorblind. None of them are colorblind. And then sample 4, there are 3 possible genotypes and phenotypes for wing color in a species of moth. Big R is red. Wings, big R, little r is orange wings. Little r is yellow wings. What are the probable offspring in a cross between an orange male moth and a yellow female moth? Well, the mode of inheritance is incomplete dominance. How do I know that? Is because when the heterozygous form is a mix between the dominant and the recessive forms, it's a blend. There's three phenotypes now, and the middle one is a mix. It's a mix of red and yellow. So whenever traits are mixing in this sort of way, it's called incomplete dominance. There's still one gene. We're talking about wing color and nothing else. So still two alleles, monohybrid, 4 boxes. I set it up. I know the alleles, big R is red, little r is yellow, but in the heterozygous form, big R, little r gives you orange because red and yellow blend. Then I figure out the parents, again, who's crossing. Well, we look at the problem and it says that the male is orange and the female is yellow. So male orange means big R, little r, right? And then yellow means little r, little r. So we're going to cross those. Go ahead and segregate. Go ahead, put the sex cells down there. And then fertilize, down and across, down and across, down and across, down and across. It looks like we get 50% orange, 50% yellow. This is for advanced students. If you feel you're advanced, you want to go for a dihybrid. How many genotypes are possible in the offspring of a cross between big A little b crossed with big A little b little b? So what do you do here? Well, we know it's autosomal because they're not saying it's sex-linked, and anytime they don't tell you anything of what it is, just figure it's autosomal. Straightforward. We know it's two genes. How do I know? Because there's two letters, big A, or sorry, just the A and then B. A and B are used, so two genes now. 4 alleles, dihybrid, 16 boxes. The alleles, again, are big A, big B, little b. And then we figure out the parents. So they already gave us the parents in the problem. And that was already stated there. That's easy. And then this time, because there's two genes, we don't just segregate. We have to do FOIL because of the law of independent assortment. So we go ahead. We know how to do that. Big A, big B, big A, little b, little a, big B, little a, little b. Do FOIL over here for these this parent. Now we have sperm and egg. Put the sperm along here. Put the eggs along here. And then go ahead and fill it out. This time keep all the A's on the left side and the B's on the right side because of alphabetical order. And then you go ahead and fill out the Punnett square. And pause the video if you want to see some of the steps. I'm just going quickly because of time. And then we go to see how many genotypes there are. Well, that's one type of genotype. Here's another kind. That's another kind. That's another kind. That's another kind. And that's another kind. Notice I didn't circle these because these are similar to that one. These are similar to these. So in reality, there's only 6 different genotypes.